Welcome to InterSTEM's Research Symposium. That is the wrong day. It's July 31st. We decided to push it back one day due to delays in research, but we're excited to get started and see what our InterSTEM summer research participants have produced throughout their 5 weeks of research. First, some introductions on the various teams and participants that we had. We had 4 main categories: biology, cybersecurity, mathematics, and physics. The most popular group by far was biology, which typically is the case with the InterSTEM Research Program. And Gordon, I believe you'll be presenting on your project soon enough. But why don't you go ahead and get started?
Okay, looks like participants can now see the screen. Is that correct? Okay.
Yep,
So hi everyone, my name is Rajvi. I'm a high school senior. I'm excited to be discussing my research paper titled Popular Browsers and Online Tracking, a Cybersecurity and Privacy Analysis. Oops. Okay, let's go all the way back. Okay, so to begin with my research topic, first, what is my research on? I focused on the online tracking that comes with today's browsers. In my paper, I used 2 tools, Cover Your Tracks and Browser Leaks, to gather data on how much information each browser tracks, such as the bits of identifying information that can be used to track a user, and I concluded my findings. My goal was to help my audience understand the browser privacy landscape and utilize proper cyber hygiene in their digital lives, which is why my research is so important and relevant as well. Okay, I now wanted to go over background information relevant to my research. Firstly, when users are online, like when we are browsing on websites, we're shopping, we're listening to music on YouTube, there's a lot of online tracking that happens with third parties and ad companies that want to know what users are doing so that they can better target ads and essentially make better products. But essentially what that means is data about a user is shared and sold, and in the majority of the cases, this happens without the user's knowledge. Since information about the user's browsing and online activities is often shared through users, it's hard to overcome this online tracking. From a privacy perspective, this is problematic when you consider that such tracking is also placed on children and minors. So here are some examples of information that can be leaked, including your IP address, geolocation, as well as browser fingerprint information, which includes things like the cookies on your computer. Let's now cover each in more detail. So talking about IP address, if it's shared, it's a privacy hazard since it allows third parties to associate the user with the various networks used. For geolocation, the browser receives latitude, longitude information of a user's location, so the browser often figures out where the user is. Now, that's okay when the user gives permission, but oftentimes the user does not give permission. Additionally, other data about the user is also shared, like what operating system they're running, their cookies, the plugins they have installed on the computer. And when all of this data is uniquely combined, it actually uniquely identifies a user. And then third parties are allowed to target specific ads to the user and also track the activity of the user across the internet, which is a breach of privacy. And the term for this is actually known as browser fingerprinting. And in my research, I took data that directly measured how unique a browser fingerprint is depending on the browser used. Okay, so the steps that I followed were I had each browser tested against the tools and I recorded the results. And I tested Firefox, Chrome, Edge, and Opera. My first tool, Cover Your Tracks, would simulate tracking domains, and it gave me feedback on different metrics, such as user agent, browser plugin details, hashes of Canvas and WebGL fingerprint, and more. And each of these metrics was actually in my research to identify how much a user could be tracked online and therefore have less privacy. In the paper, I said that the higher the bits of identifying information there were, the less privacy friendly the browser was. So after running the test for each browser, I would get to see how many bits of identifying information or data that the browser collected. And in the picture in the right, you can see how a metric would have its own set of data. And this is just a sample. There were many such metrics used. I aggregated all of the metrics for each browser into data tables for analysis. Moving on to test number 2 with browser leaks, I chose to do the geolocation API test, as you can see in the image over here. Now, how does the tool work? According to the site, it checks for browser permissions for geolocation, and based on the latitude, longitude, and accuracy received from the browser, it shows the radius of the location of a user on an interactive map. This is a snapshot of what the results would look like with this tool. It would actually figure out the address of the user, and it would tell you with what accuracy it thinks it has guessed, along with the range on a map. So the accuracy changed with the browser you open the site with. And for the paper, I decided that a more precise location was more troubling. A higher accuracy hints that more intrusive detection of a user's location is possible, and less accuracy was more desirable for privacy. All right, so what were my findings? With Cover Your Tracks, the data shows that with all 4 browsers, the user will have a unique browser fingerprint. And the more unique the fingerprint is, the more trackable a user is. So it's worse for privacy. However, Firefox was the only one that provided partial protection against tracking ads and invisible trackers. However, the other 3 did not. But when I compared additional metrics for privacy, I found the results for how many bits of identifying information were for each browser, such as user agent. And when considering user agent, Opera had more bits at 10.95, whereas Firefox had the least at 4.296. And again, more bits means less privacy. Depending on the test, however, browsers did differently. For example, for the metric hash of Canvas fingerprint, Firefox performed poorly compared to all others, and Chrome actually performed better in terms of privacy. Chrome was at 6.98, but Firefox was at 13.01. Then again, for some tests, like with the browser plugin data, often the browsers performed very similar to each other. Here, 3 out of 4 browsers had the same value, which is 1.15, and the 4th data point is very close to the others at 1.14. And I ran a lot of these tests. For example, the HTTP accept headers data, cookies, WebGL fingerprint, and so on. There were around like 13 tests, and I collected it all into this chart over here, where I tallied up the number of tests that a browser performed better than the others. Moving on to the second test with browser leaks, in terms of greater accuracy to lesser accuracy, it was Firefox, then Chrome, then Edge, and Opera. And however, like in terms of privacy, coming in last is actually preferred. So in terms of this test, more privacy friendly would be Opera all the way down to Firefox. The data visualization as a radial bar graph can be seen here. Note how much the difference is with Opera, which was better for privacy, which you can see here in blue, compared to the others, which were more or less the same. Finally, what was the conclusion? In my research, I found that each browser offers varying levels of privacy protection depending on the test you're doing, what metric you're looking at, and what is more important to you as a user. I hope that through this presentation, you were able to gain some insight about the complexities associated with browsers, online tracking, privacy, and ultimately cybersecurity. For the next stage of the research, to take it further, I want to test on different device models. My device, my data that I gathered was a Windows 11 device, which I use to run the different browsers, but I'm curious to see about how and whether or not the data will change if I test it with a Linux device, for instance. Okay, reflecting on the whole process, it was a great experience and my first time doing research. There were definitely a lot of obstacles, but each time I managed to come up with a solution, which I think is what the scientific process is all about. First, there was a challenge of the topic selection since I had another topic in mind when I first began, but it was very hard to do testing and kind of trying to figure out how I would do the data gathering about it. So after a lot of thought and kind of trying to work things out, I decided to work on research with the topic of privacy and browsers. Another hurdle was managing to test and even choosing the attributes to focus on because I had to define my own metrics and clearly define them and narrow them down. But I'm happy with the methodology I used in my research since it came to me after hours of brainstorming. And I also wanted to do original research, something that I wasn't sure if that was possible. So I really was satisfied to be able to manage that. And finally, just like coming up with the data visualization and kind of wrapping up with a big conclusion. And overall, I had lots of fun doing my research and I learned a lot along the way. And with that, I wanted to say thank you for listening.
Thank you so much, Rajvi. I'm glad you enjoyed this experience, and your research findings were fascinating. I had no idea all of that about the privacy concerns with our tabs. So thank you for
Yay.
teaching us about that. All right. Now Gordon will present on marine biotechnology. Excited to hear more about this, Gordon.
Okay. First of all, hello everyone. Once again, my name is Gordon Chang, and I'm about to be a high school senior within Southern California. So yeah, just getting started off with our introduction. So yeah, a huge issue within our society today has been marine degradation, and it's been a huge issue that many people have debated, and it adversely affects a lot of marine ecosystems, and it indirectly affects human life. So, and the reason for, a lot of the marine preservation that we have, a lot of the efforts that we have is because people say that, the ocean accounts for a large proportion of our food supply, which is completely true. And it is a huge reason for efforts to support funding for marine preservation. But one additional thing has been the field of marine pharmacognosy, which is essentially the field of using various techniques and marine-based applications to find cancer treatments. And within this research project, I focused on cytarabine, trabectin, and as well as a couple marine-based peptides. So within our methods, so at first I looked at several research papers regarding cytarabine, which first is marine drugs for cancer, surfacing biotechnological innovations from the oceans, which is an introductory research paper towards various cancer treatments that have evolved from the ocean, as well as a more in-depth review on cytarabine. So this was originally sourced from the Caribbean sea sponge, and research was done on its molecular structure of these organisms and how that potentially helps with cancer treatment. And with trabectin, now this drug was actually sourced from the Caribbean sea squirt, which for those of you that don't know, kind of think of it a little bit like a sea cucumber. But either way, its direct usages and side effects were examined. as well as an in-depth review on trabectedin and marine drugs for cancer. Once again, that same research paper, as stated before, was used to discover more about the process that this works. And so here are some diagrams on, and this is cytarabine and its different functions, as well as how it kind of destroys the DNA structure. And this is likewise cytarabine. So in terms of our results, yeah, so cytarabine has two nucleosides of interest. And for those of you that don't know, nucleosides are essentially the nitrogenous base attached to the sugar. So it's opposed to a nucleotide. Now, through kind of the very rather complicated conversion process, these nucleosides will be incorporated into the DNA and to kind of like substitute for cytosine. But because, obviously natural DNA has cytosine in it, and now that this has been replaced, this will be kind of halt the process of DNA synthesis. And as a result, once these cells reaches the first growth phase, apoptosis will occur. And this is extremely beneficial within tumor cells, as those are the cells that scientists and researchers are trying to eliminate. And same thing, a desired artificial apoptosis is what trabectedin aims to do as well. And in this scenario, trabectedin binds to the minor groove of DNA. This leads to changed shape in DNA, which further leads to the disruption of the cell cycle. And this will ultimately lead to apoptosis as the cell notices kind of like some errors within its structure and everything like that. And furthermore, there have been studies done that shown that trabectedin is kind of shown to reduce the immune response of the tumor microenvironment, TME. So the tumor microenvironment is essentially kind of like that area near the tumor cells around it, and it is largely responsible for growth. And so obviously efforts to kill those cells have been very difficult, as those cells have immune systems as well. And so this trabectedin has been a tremendous help in the sense that it can kind of ignore the immune response of those tumor cells. All right, so in terms of our discussion. So yeah, the results of this study do demonstrate the potential of future funding for marine research. And trabectedin have been discovered as potential treatments for cancer as they disrupt cell cycles and can therefore enable artificial apoptosis. So yeah, this is definitely going to be adding a further layer of complexity in terms of various reasons to reserve federal funding for marine preservation. And this study was limited as the only resources available were previously published articles. And given the various personal circumstances that I was dealing with, as well as the time given to complete this research project, the study was not done with maximum details. And in terms of our conclusion. So yeah, overall, the purpose of this study was to analyze cancer treatments with a marine origin in order to demonstrate the need for funding to preserve marine habitats. And the results of this study are a clear indication of the potential of marine resources. As the ocean has been left largely unexplored, further research into marine life, especially organisms that live in more variable ecosystems, could give more insight into treatments for cancer. Especially, I think there has been research done on organisms that live within deeper vents that have very different ecosystems that we do, and therefore could potentially provide as a source of further research that can help within the field of biotechnology. And a future direction of this study would be to determine how to mitigate the side effects of these cancer treatments and sorry, I forgot to complete that, and therefore make it more efficient. Because, for example, cytarabine actually has some, pretty severe side effects, and these include nausea and, as well as rashes and ulcer formations. So that is why further study needs to be done, and it is also why these marine treatments haven't been as widespread as some may hope. But nonetheless, my research presentation was a lot shorter. So thank you all very much for listening.
Thank you so much, Gordon. I'm glad you were able to finish it. I know you had some issues going on, but the final product turned out amazing. And like Rajvi, I was thoroughly impressed by that. I had no idea that marine life could be used to help with cancer treatment. So that was very fascinating to learn about. And now I'm going to go through some of the other participants in the research program and their research questions that they had begun and will continue to be working on. So first is Arshi with mathematics. And they were looking at what are BMI related errors and inaccuracies as a measurement of obesity and overall health. Next is Mariana with physics and looking at how do massive bodies influence astronomical observation. I believe she was looking at a specific star in particular, but I do not have it in the notes here, which, but I'm sure that one will turn out interesting as well. Zainab with biology, again, our most popular group. Again, same research question, but that's okay. Then Deborah was also with biology and looking at what are the mechanisms that lead to metabolic impairment of brown. I'm not going to attempt that word during aging. So again, another biology topic. And then finally, Jayden with biology, looking at the risks of autoimmune disease after the usage of checkpoint inhibitor immunotherapies. So congratulations to all of you for completing the InterSTEM Summer Research Program, and thank you so much for participating.